Good morning, everybody. I'm happy to introduce our first speaker today. Dr. Kevin Begcy is an assistant professor in the Department of Environmental Horticulture at the University of Florida. His research group focuses on understanding plant developmental responses to biotic and abiotic stress. His overall research goal is to elucidate novel pathways and mechanisms that could be used to improve crop performance under unfavorable environmental conditions. I'm excited to hear about how this research relates to our overall theme of breeding for climate change. So Kevin, the floor is yours.
Thank you, Nadia, for the introduction. So let me share my screen with everyone. Can you see now?
Yes.
All right, perfect. First, I would like to congratulate the organizers of this fantastic symposium. I think it's a great opportunity for discussing great science and networking with different people in the industry as well as in academia. So today I'm going to be sharing a little bit of what my group have been doing in the last probably 3-4 years, studying stress responses, particularly in maize. So I would like first to acknowledge the fantastic group of students that have been conducting this research. The data I'm going to be presenting today mostly comes from Shinli Li. He's a 4-year PhD student. Andrea Geza, he's a 2nd-year PhD student. And Katie Chase, she's a 1st-year master's student. So if we were in a in-person symposium and I asked the audience whether you have been stressed lately, I assume the entire crowd will raise their hands. So can you imagine what happened with plants where they are either in the wild or they are in the field, experiencing normal changes in temperatures, normal changes in continuous seasons, plus pathogens and diverse abiotics as well as biotic stresses. So how plants cope with these constant changes? So this is one of the topics of our research, and the approach that we take, we mainly are focused on reproductive development. So even though there are many important stages during plant development, we are interested in 3 main components: the female gametophyte is here, the male gametophyte, and the seed development. So the data I'm going to be sharing with you today mostly comes from our results in the male gametophyte. And for that, we have been using a particular interesting crop, maize, as a model. Maize arguably is one of the most important crops globally. It has a really well not annotated, but well sequenced genome. Lately have been new updates on it. But during this climate change situation, we have seen that so we have seen that in the last probably 100 years, temperatures have fluctuated in a global scale. We have seen regions where temperatures become 1 or 2 degrees centigrade above the threshold. Others during the winter time becomes really cold. So we have seen this climate change varying across the entire planet. So that have imposed a particular challenge on crop production. So in order to see how those changes impact crop productivity, as I said before, we use maize, and this is the results of a picture of some of our plots in, I think it was 2017, where we saw a really dramatic decrease in yield. We see a few seeds in some crops, others we saw kind of halfway through. But then we were interested actually what is causing this particular phenotype. When we look at the male phenotype, this is something that many of you have seen, is that you will see pollen grains that are fully formed, which means that they are viable, the ones that are really dark here. And you see also pollen grains that are kind of unviable, the ones that are more light color here. So those particular pollen, they don't germinate. What that means is they are not able to transfer the genetic material from the male side to the female side during the fertilization process. So many studies conducted, this is something not new. We have seen that across many plant species. We see, for instance, examples in brassica, populus, beans, where heat stress basically impacts pollen development. This is something that have been discussed for many long times. Scientists, even in 80s, 90s, they already realized that particular problem. Even though this is not novel, the research in my lab takes a different approach. So rather than looking at the broad context, how heat stress impacts the entire overall reproductive development, we are interested to see how are the impacts in every single developmental stage. And the reason why we are looking at developmental stage responses is because we know that there are tissue-specific gene expression, tissue-specific protein expressions. We see that every developmental stage is different. So in order to see how plants respond at the developmental level, we wanted to look at how each pollen stage have been impacted or is impacted by heat stress. So here, this is a normal pollen development transition. So it starts with pollen model cells, which then goes to myocytes, then tetra stage, unicellular, bicellular, tricellular, and then mature pollen, which is the one that germinates and then fertilize the ovaries. So this can be divided in microsporogenesis and microgametogenesis. One of the main problems in studying developmentally the impact of stresses is how you are able to dissect only the developmental responses rather than the entire response of the plant. So in order to do that, we first develop a method that allows to systematically isolate pools of individual pollen stages during maize plant development. So we use the well-known leaf color method, and we try to correlate it with the different stages of pollen development. Even though we didn't find a 100% correlation, we were confident that the amount of pollen that we can isolate it were from an individual pool or individual developmental stage. This particular study was conducted in B73 imbrine line. We tried W22, A188. The results are not as consistent as B73, but that's something that we can play around, and that can give us good number of isolates in individual developmental stages. So for instance, you can see here in B13 that most of the pollen is in the unicellular stage. Again, we use the central spike of the tassel, so we were just concentrated to have a unique pool of cells in the same developmental stage. And you can see here that basically most of the cells that we isolated were in the same developmental stage. Here is the tetra stage, which means we use mostly V12, actually V12 maize plants, and here we use, this is bicellular stage, and we use mostly B15 plants. So after we have developed that method, we went and then look at every individual developmental stage. I will be sharing mostly two stages, tetra stage and bicellular stage, even though we have conducted research on almost all the stages. Because of time, I'm going to be sharing the only tetra stage and bicellular stage. So bicellular stage is a really important stage during pollen development, since it's a transition between the microsporocyte to the megasporocyte. So one thing that happens before that transition is that at the tetra stage, you can see here, there is no starch accumulation. So this is previous starch accumulation in the pollen development. Why starch is important for pollen development? Because that's the one that basically going to produce, helps with the energy production for later germination. So we particularly focus on heat stressing plants that were at the tetrad stage of pollen development. Here is just a scheme showing how we did that particular study. We heat stressed it here at the tetrad stage. You can see in the red line. And we also kept a group of plants as a control, and you can see here in blue at normal temperature. We conducted physiological, biochemical, omics technologies to see how actually the plants responded. And we collected samples right after the stress, during the stress, as well as later during normal development. Something that I wanted to point out is that after we applied the heat stress, which basically lasted for 2 days at 35 degrees, we placed the plants back into normal conditions just to see how that particular stress only during the tetrad stage may have any impacts on normal pollen development. To our surprise, that transient stress of 2 days impacted greatly the shape of the tetrad stage. You can see here on the top, a non-stress pollen. You see the round type of shape of the tetrad stage. However, the heat stress ones, they clearly were a little bit shrink, and that one was something that caught our attention. Since we know that pollens are not individual entities and they are maintained in capsule within enthers, we also look at an enther development. Even though enther development is something that have been really well studied, we just wanted to see whether the stress that we applied actually caused similar phenotype I've described before. And we look at here at the tetrad stage, maculated microspores, bicellular stage, tricellular, and mature tricellular pollen. And here on the top, we have non-stress conditions. On the bottom here, we have heat stress conditions. And 3 important components stood out that were different. So we see here that the epidermis cell layer was degraded a little bit slower in the heat stress samples than the non-stress ones. That caused a little bit of delay of the releasing of the pollen. Also, we observed a degeneration of the tapetum. It was a little bit slower again on the heat stress samples, as well as endocytium. It is interesting because even though we apply heat stress, many have reported that the tapetum degrades faster, at least in maize and in the inbred line B73. This particularly transient heat stress actually delays the process of tapetum degradation, which is really important for the pollen formation. Another important observation was that you see here the mature tricellular pollen in non-stress one was really strong, a strongly intense stain, which is an indication of pollen viability, something that we didn't observe here in the heat stress ones. So after we analyze right after the tetrad stage, and when we stop the stress and then we put the plants on their normal development, we look at one important trait for pollen, which is pollen germination. So first we did an in vitro assay, which basically consists on putting the pollens on a media that allows pollen to germinate. We provide all the components needed for the pollen to germinate as if they were in the plant. What we see this in vitro assay shows that many of the heat stress pollen didn't germinate, as you can see here in the white arrows. A few, as normal, in the non-stress one also didn't germinate. So when we quantified that particular response, we observed that around 80% of the non-stress pollen germinated, and around 20% of the heat stress ones also germinated. For the people that work with maize, you will say, okay, 20% of pollen in maize, because of maize has a lot of pollen, that maybe doesn't have a really strong impact on fertility or seed set. But before moving to that, we also did an in vitro assay. So in vitro assay, basically we use normal silks, we place non-stress pollen and heat stress pollen. And in the non-stress one, we see how the pollen tube germinates normally. So basically germinates, penetrates the silks, and basically goes all the way down to reach the ovule. In the heat stress ones, the pollen is able to stick, a required process for pollen germination. But even though it is able to stick into the maize silks, the germination didn't happen. So we went further and analyzed to see what are the effects of seed set. And what we see here is our controls. So basically non-stress pollen and non-stress cups, we see full seed sets. And basically here we use non-stress cups and heat stress pollen. And we see a diverse type of response. Some cups were more full than others, but again, in general, we see a strong reduction in seed set, basically because of the failure in fertilization. So we wanted to understand better what was the cause of this particular phenotype. And then we started biochemically checking on pollen, mature pollen after being stressed at the tetra stage. And we first look for a starch, which is one of the most important components, as I said before, for pollen germination. And this is a non-stress pollen, and this is a heat stress pollen. You see partial accumulation of cellular components. So basically starch were just reduced. And when we quantify it, basically we see a significant reduction between non-stress and heat stress ones. Same happens for a technique called FDA staining. FDA is a enzymatic assay that allows a chemical component of FDA to penetrate the pollen. When the enzymatic activity is really strong, you see a strong signal, as we can see here, fluorescent signal. When the enzymatic activity is low, basically you see a decrease in the fluorescence. When we quantify, also we saw a significant difference between the heat stress one and the non-stress one. So this one was the first indication that the starch as well as enzymatic activities were impacted. So we went and did a transcriptomic approach. We use RNA-seq Illumina analysis. As you can see, we found many genes that were disrupted by the heat stress at the tetra stage. And again, I would like to reinforce that these particular samples were took at the maturity. So first we use cellular markers as expected, heat shock proteins, which are important for markers as the heat stress markers. We observed that even though the stress happened in the tetra stage during the recovery process, those genes were still upregulated. So even though the stress happened before, at least this particular inbred line was not able to recover again to the normal stage. We see a really high expression of those particular markers. So then we look at exactly what was the responses, what happened, why this particular pollen didn't recover. And we found a lot of genes that are important for fertilization. For instance, kinases are known to be important for the fertilization process. We study a few of them. We saw upregulation and downregulation, and then we perform a network analysis, and basically a few of them work together with energy pathways as well as the heat shock protein responses. So we went further and look at exactly at the biochemical pathway that were impacted, and among the different ones that were impacted, we look at two main important ones for the pollen germination, which are the energy biosynthesis pathway and the lipid biosynthesis pathway. As you can see here, the enzymes, or also the genes in red, are upregulated. The ones that are in blue are downregulated. So first, we're going to be looking at this particular energy biosynthesis pathway, and what we saw was that even though, as you could remember, the starch was reduced, and the enzyme that basically converts starch into glucose also was reduced, ADP glucose. However, plants didn't basically stood there. So in order to overcome this particular reduction in the amount of starch, maize pollen used another pathway. So they use the invertage, which basically converts glucose into sucrose into glucose and fructose. So this, even though this particular pathway was diminished, plants basically turn all the hope into the invertage to basically be able to produce enough energy. And also was another gene and enzyme were upregulated, the phosphofructokinase. So this one was shut down, but this one was increased here. However, at the end of the pathway, we saw also a reduction. Phosphate dehydrogenase as well as enolase, important for the conversion of this particular glyceraldehyde into pyruvate. They were downregulated. What we observed was at the end, ATP synthesis was decreased in the heat stress pollen compared to the non-stress one. So in order to prove that not only at the transcriptomic level, that was the case, we performed metabolomics, and we actually confirmed our transcriptomic results. So glucose is upregulated, was upregulated here in heat stress samples, and this upregulation basically comes from the invertase activity, not because of the ADP glucose activity. Same fructose was upregulated, as we can see here, both. And then pyruvate levels were slightly reduced in the heat stress compared to the non-stress one. A similar observation was in the lipid biosynthesis pathway, where basically lipids are really important because they are essential components on the newly membrane formation during the pollen tube growth. We observe upregulation of the 3-oxacyl synthase as well as the glossy one. However, acyl carrier desaturase was downregulated. So our hypothesis is that this particular gene was the bottleneck, even though those was upregulated. This particular gene was not able to convert to the kinol into the different lipid compounds. And basically, when we perform metabolomics, we observe a reduction in the heat stress samples in all the different lipids with different carbon compounds, C15, C16, C17, and C18, which are all of them fundamental again for the cell wall formation, cell membrane formation. As a model, we propose that basically when heat stress happens at the delta stage, we see an increase in sucrose because of the activity of the invertase, which increase the excess phosphate pools. However, the starch, which is also another contributor of the phosphate pools, basically was decreased, and therefore because of the activity of the ADP glucose. That one resulted in decreasing pyruvate, fatty acids, and energy, which basically impacted the pollen germination as well as fertilization. So that's one initial response that we observe at the tetrad stage. So heat stress at the tetrad stage of pollen development causes male sterility. And this is what basically because of two important, the heat stress targeted two important biochemically important pathways, energy and lipids. But then we shifted our attention to the bicellular stage. We were expecting similar results, and we used the same leaf color method as a way to collect the pollen. And one thing that actually took our attention was that the heat stress pollen at the bicellular stage didn't respond as expected. We didn't see any effects in the pollen germination when they were mature. So we did in vitro assays, and we also did in vivo assays. You can see the pollen grains germinating normally here in the controls or non-stress samples versus the heat stress ones. Same happens here in vitro. So this is something we repeated multiple times, getting the same results. We then decided to look at more in detail. And here you can see starch composition. They were no different. We used DAPI staining. Basically, DAPI is a staining that allows coloration of the DNA within the nucleus. Those were in the same developmental stage. We didn't see FDA staining was no difference between heat stress and non-stress. We use another staining, Alexander staining, which also stain for starch. We didn't see differences. We check, okay, if the difference are not, maybe the germination speed of the pollen tube is different. No, we didn't see differences in germination, in the speed of the germination, the pollen tube. Also, the pollen germination, when we quantified, we didn't see any difference at all. Same for pollen tube length. So at this point, we were, okay, maybe bicellular stage is resilient to stress. So when plant development already reached that stage, is not relevant when the heat stress happens. Maybe the earliest stages are more susceptible. And then what we did, we just cross heat stress pollen at the bicellular stage with non-stress cups. This is our control samples, full seed set. But again, we observed the same similar phenotype. So that was a really contradictory result, because if we didn't see anything happening here within the pollen and the female side was not stressed, why we still see a failure in the fertilization? So in order to look at and try to understand that particular observation, we study, we propose we look at a different hypothesis. So again, this is a normal pollen development stage. We have tetrad, unicellular, bicellular, and tricellular. One of the important stages between the bicellular to the tricellular is the formation of the sperm cells. This is the vegetative nucleus, this is the regenerative nucleus, and then after this transition, they separate, forming the two sperm cells. One is going to fertilize the embryo, the other one is going to fertilize the central cell to form the embryo, the other one is going to fertilize, going to form the endosperm. So our hypothesis was that basically there was no formation of a second sperm cell. So basically there was no separation of the regenerative nucleus forming the two sperm cells. And that's why basically there was no fertilization at all. That's basically explained here, where after heat stress, you need to have a formation of two sperm cells, which need to travel and then fertilize. Our hypothesis was that only impair pollen travel, and because they were not two particular sperm cells developed, we observe a failure in fertilization. In order to confirm our hypothesis, we constructed a marker line. So we used a YFP marker line. We knew that we have a few genes that are only expressed in the sperm cells, and we fusioned that particular gene with the YFP to visualize the particular expression. When we had that plans ready, we perform a heat stress experiment. On the non-stress one, we see here, this is basically the sperm cells. You see a nice spindle formation. So after the bicellular to the tricellular, you see a division of this particular sperm cells, forming two individual sperm cells. Clearly here, this is the intermediate stage. In the heat stress ones, we didn't see that particular clearly. It was kind of affected that process of separation of the sperm cell formation. So, okay, that one match a little bit with our hypothesis. However, when we look at in detail during the pollen germination, we observe that indeed the sperm cells, they are able to travel, and we observed two of them. So our initial hypothesis that the sperm cell formation was impacted was actually not true. What actually happened was that the heat stress samples kept the sperm cells inside the grain. So the genetic material never traveled out into the pollen tube. That's why the fertilization was impaired. There was no genetic material traveling to the pollen tube, so the fertilization process was particularly not happening. Nothing in contrast with the non-stress one, where you can see here the two clearly sperm cells traveling across the pollen tube. So in order to understand better that, we isolated sperm cells. So we use a technique called a Perkel gradient. So we use different concentration of Perkel to isolate only sperm cells. Many studies have been conducted with pollen, but they use entire grains, which basically does not provide information regarding specificity in the sperm cells or the entire pollen grain. So we decided to do only sperm cells. We isolate individual sperm cells, as you can see them here. And then we perform again a transcriptomic analysis. And we observe around under heat stress at the bacillary stage. We observe around 600, almost 700 genes totally misregulated. Around 478 went up, around 200 were downregulated. Here is just a heat map showing a few genes up, a few genes down. We observe, we perform gene ontology analysis, many genes up, many genes down. At this point, and as you may imagine, many of the studies base their decision on designing or to study candidate genes on only transcriptomics. So we didn't want to base our decision only on transcriptomics. So we also performed sperm cell proteomic. What we found was that there were around 1,400 proteins that were common on both, and around 600 were unique for non-heated, non-stressed pollen, and around 1,300 were unique in the heated stress pollen. So we crossed the proteomics with the transcriptomics, and then what we did, we picked samples, genes that were present in both data sets, not only in the transcriptomic, but also in the proteomics. Since maize transformation takes quite a long time, and we didn't want to risk of studying genes that maybe are not the target of the heat stress, or genes that were not actually giving the response of what happened during the sperm cell. We first perform in situ hybridization. So we studied initially 2 genes, and the in situ hybridization allows us to basically confirm where the stack expression of those particular genes within any given tissue. When we look at the enters, we see a clear expression, as you can see, a strong signal here in the sperm cells of both the gene 1 and the gene 2 target. Then we perform also transient expressions. First, we use tobacco pollen transient transformation to see whether those genes were indeed expressed in the sperm cells. We see strong expression in the here in the sperm cells of tobacco pollen tube, as well as in the pollen grains. We also did transient transformation in leaves of tobacco plants, and they were again strongly expressed in the nucleus, an extra confirmation. But then we also use BMS suspension cells, again, to double check that those particular genes are expressed into the nucleus. We wanted to be sure before embarking in a transformation process to be sure that our genes were actually the targets. So we see a strong expression again in the nucleus. So we decided to go further, and we perform CRISPR. We designed CRISPR for those 2 genes. We use 2 different set of constructs. We use in the same construct, we target both genes in order to maximize the chances to have a double mutant of both genes. And the reason why we did it with both genes, because those genes were genes belonging to the same gene family. What we found a myriad of different transformants. We found single mutants. We found most of the transformants that we got were single mutants, so we started the process of crosses. Interestingly, we found genes that plants that were edited with around 50 base pairs, and another plants were edited around almost 900 base pairs. So we got plenty of plants. We started genotyping all of them, and this is how they look like. During vegetative development, they look normal, nothing different than any wild type plant. So at this point, what we are in the T1 generation. And this is just wild type transform. And these are the 3 independent CRISPR-Cas9s. What is important, we have heterozygous of double mutants. And what you can see here is that at the tricellular stage, you will expect to see the 2 sperm cells and the vegetative nucleus. And see here, you can see here the 2 sperm cells, vegetative nucleus. Something that is really interesting for us right now, we observe, you see strong signaling all over. So the function of that gene is involved in several components during the replication process. And interestingly, the sperm cells are not clearly formed. So basically, you don't see a particular condensed sperm cell formation. You see a dispersed signaling all over. So our hypothesis initially is that maybe that particular 2 candidates are important for the sperm cell architecture, for the chromatin architecture, and that's why they are just basically, when they are not present, the structure cannot be maintained, and then the genetic material is dispersed all over the pollen grain. We also look at pollen germination, and you can see wild type transform. You can see here the normal germination of the pollen of the pollen tubes. However, in the mutants, the double mutants, what we observed was that they start to basically not growing as here. You see straight pollen trying to find, the ovule. Here they start to growth, and then they start to basically curl a little bit, and then it is a really strange change in orientation, something that is not normally observed in the wild type pollen. So now we are quantifying the differences between that particular phenotype to see whether this is something that is related to the 2 genes that we were looking at or something else. And then we also look at the fertilization rate, C-set, and we cross them with B73, we cross them with A188, we are also crossing them with W22. This is here the wild type. We started seeing a few reductions in some of the crosses. However, this is not conclusive, since still we are working with heterozygous plants, and they are in the T1. We are in the process of getting homozygous plants in the T2 generation. After we confirm that they are homozygous, we're going to perform the heat stress analysis and whether those particular 2 genes are critical for the response to the heat stress, as well as the sperm cell formation. So in summary, and what is the take-home message is that the impact of heat stress on pollen development is different depending on the stage. So it's not the same that when you see a stress happening, if we go to the vegetative development, what happened during the seedling stage, that during before flowering transition. So every developmental stage is different and has a myriad of responses. So in the case of pollen development, is different depending on the stage. So the third stage is susceptible, and basically the susceptibility comes from the impact on lipids and energy biosynthesis. The bicellular stage is a little bit more resilient, seems to be, but then at the fertilization stage, the fertilization is really impacted, which means that the responses or the molecular pathway that are affected by heat stress at the bicellular stage are different than the tetrad stage. So with that, I would like to thank everyone, and I will be happy to take any questions.
Thank you, Dr. Begcy. We have a question from the chat. From the results of your work, we can see that high temperature affects pollen negatively and hence causes significant yield decrease. In the current climate change scenario, do you think that the development of heat tolerant lines using some of these genes that you were working on for pollen development should be one of the main focuses for maize breeders?
That's one of our objectives. So we want to find critical genes that help breeders to have more resilient pollen. So pollen that even though heat stress happens, there is enough pollen for the seed production. I didn't show here, but we have also other candidates that basically the amount of pollen that they produce is a seed way more controlled. So basically we have twice the amount of pollen than the control plants. So that has a really nice potential implication, because if you increase the amount of pollen, even though the stress happening, a good amount of them gonna have good chance to fertilize properly, and therefore you're gonna have a normal seed set. So we are working on those candidates to see whether they perform really well in the field. So far, the data we have is data in the greenhouse, but we are now transitioning to the field to see whether the performance is as observed in the greenhouse.
Thank you. I have another question from Dr. Narakuduti. She asks, what is the hypothesis for the slow degradation of the tapetum and the epidermal layer under heat stress? Why do you think? Yeah.
That was the first study, right? So in this case, what we think is that because of our stress was only for 2 days, it was not long enough to cause what many people have observed, a rapid degradation. So I think these two stress, basically what it does is just mess up entirely with the molecular responses, and then the plant didn't know exactly what to do. And basically what happens, the tapetum started to slowly degrade, just to release, just to acclimate to the stress conditions, and that's what we see the differences.
Do you think that the disruption of those cellular structures, the tapetum and the epidermal layer, also contributed to them not being able to germinate well?
That's really an important stage. So the tapetum is known that basically nourish the pollen. So there is a critical developmental transition where the tapetum start to release all the compounds that will nourish the pollen. If that nourishment process is affected, as you may imagine, for instance, what happened with small babies, if they are not properly nourished, the normal development is going to be impacted. So same happens with pollen. If the normal nourish process is affected, the pollen doesn't want to get the biochemical and physiological, also the molecular response that is normal expected during pollen development.
One last question that I have you considered doing overexpression lines so that on those replication genes that you spoke about, maybe this will create more pollen?
That's something, the one that I talked, we have overexpression. So basically the one that produces more pollen is our overexpression line. The one that has have a CRISPR, the gene having CRISPR basically reduces the amount of pollen. So we see a really dramatic change when the gene is overexpressed and when the gene is not present. So it's a really promising, but still we are in the data in the preliminary stage where what we saw was only in the greenhouse. We want to be sure that it actually happens in the field.
Thank you, Dr. Begcy. I really enjoyed your talk. Those pictures and the videos are super cool. Very, very nice. I will now we'll take a short break. And when we come back, we'll have the 3-minute flash talk competitions with students. So everybody should expect to come back around 11. 11 a.m. So we'll have a 15-minute coffee break. Thank you, everybody.